And so now I want to finally discuss how contigs can be assembled into what are called scaffolds. And so scaffolds are basically arrangements of contigs based on other assays that you can do in order to figure out which contigs follow which other ones in the genome. And so, nonetheless, either through this overlap layout consensus approach or, one of the kind of more modern assembling approaches, at the end of the day, assemblers are very good at kind of handing you back a set of contigs. So those are, again, stretches of DNA sequence that assemblers are relatively sure form one continuous region of the original genome. But, a lot of these assemblers basically leave it up to you to actually figure out, okay, if my assembler comes back and gives me 4 contigs, what is the order of these contigs, the relative order of these contigs in the original genome? And so there's basically 3 approaches that we'll talk about in this lecture to kind of scaffolding these contigs or figuring out, what is the relative order. And so the first approach is through mate pair sequencing. Second one is called optical mapping. And the third is through chromosome conformation capture, which we'll actually cover in a future lecture, but we'll go over again here just because it's relevant to assembly. And so in the mate pair genome assembly approach, basically you'll recall from the previous lecture that even technologies like Illumina SBS can give you paired end reads out of DNA fragments. And so it's worth noting that you can get paired end reads from both kind of short inserts, as short fragments as well as long fragments. And so the point of mate pair genome assembly is that it takes advantage of the fact that oftentimes, even if you're only sequencing the ends of a given fragment, you oftentimes know, either because you did some size selection during your library prep or whatnot, you oftentimes know how big the original DNA fragment was, even if you're only sequencing the ends. And so that's powerful because, through mate pair sequencing, you get pairs of reads and you know approximately what the distance between them is on the original genome, even if you don't know what the sequence between them looks like. And so why this is powerful is that, on one hand, if you use an assembler to, for example, assemble contigs in your genome, and you end up, your assembler tells you, okay, there's 4 contigs in my genome. If you have a bunch of mate pair reads which span 2 different contigs, then that actually gives you a measure of physical distance between contigs. So if you have a mate pair where one read sits in one contig and the other member of the pair sits in another contig, then because you know the distance between that pair of reads, you therefore kind of have an idea of how far apart physically the 2 contigs are. And so mate pair based genome assemblies is actually difficult in practice because it's really only valuable when you do mate pair sequencing on large DNA fragments. Because if you're doing mate pair sequencing on short fragments, then you're only able to connect contigs that are very close to each other, and those are more likely to be joined anyways. It's less likely to have 2 contigs that are very close in distance, but you weren't able to assemble, than the case where you have 2 contigs that are actually far away from each other. And so in order to do mate pair based assembly accurately, you need to be able to sequence large, paired reads from large genomic segments. And that's hard because it's hard to fragment DNA in a way such that you get large fragments and that you don't accidentally fragment it further. And the second main problem is that you actually need a lot of input starting material in order to do deep enough mate pair sequencing and get these like large fragments. And so in a lot of cases, you don't actually have enough genomic material to actually do the mate pair based assembly, the sequencing and assembly in the first place. And so another approach to determining the order of contigs is called optical mapping. So in optical mapping, you take your genomic DNA, you fragment them into large fragments, and put them on a microfluidic device, after which you take a collection of restriction enzymes for which you know their recognition sequence, and you use those restriction enzymes to make cuts across each of the genomic fragments. And so after you stain all of the DNA molecules, you basically can get a visualization of not only where are all of the genomic fragments, but where have all the cuts basically been made along each of the genomic fragments due to the restriction enzymes. And so then you have, you basically get to the more complicated part of the process, which is where you take the original genomic fragments and you try to overlap them to figure out which genomic fragments share similar sequence coming from the same region of the genome. And so how you do this is that based under the assumption that you have a lot of input genomic DNA material and the fragmentation process was random, you should essentially have, for any given piece of the genome, you should have multiple fragments that came from that part of the genome. And so here on the bottom right-hand corner of the slide, you have a hypothetical fragment 1, 2, and 3. And so obviously on the optical slide itself, you don't know that fragment 1, 2, and 3 per se came from the same region of the genome. But what you can figure out in this specific case is that even though fragment 1, 2, and 3 aren't ordered for you on the slide, by looking at the length of the segments of each fragment that have been cut, if fragment 1 and 2, for example, came from adjacent regions of the genome, then they should basically share, for example, in this case, subfragments F1, F2, and F3, because that's a region of the genome that's common to fragment 1 and 2. And so the restriction enzymes that are responsible for making the F1, F2, and the F2, F3 cut will cut the same location in both fragment 1 and 2. And so fragment 1 and 2 will have these F1, F2, F3 regions of the same size present in both fragments. And so based on this principle, you can then kind of overlap your different fragments so that you can figure out, okay, fragment 1 and 2 share three fragments of subfragments of the same length. And these three subfragments are at the end of fragment 2 and the beginning of fragment 1. And so fragment 2 is likely to precede fragment 1 on the genome. And so based on that principle, you can basically reassemble the original genome at kind of a low level of resolution, as you can see on the bottom left-hand corner of the slide. And so in that sense, you generate this genomic optical map because that basically tells you where along the genome different cuts were made due to the restriction enzymes that you used back in step 3. And so this is an example of an actual optical mapping visualization that you would get after staining. And so the reason this helps you scaffold your contigs is that, remember, you know the recognition sequence of these restriction enzymes. And so your optical map gives you these genomic fragments and the locations of where the restriction enzymes cut. And so that's represented by the blue lines and the green dots, where the green dots tell you where the restriction enzymes cut. And you also have your contigs that you know the sequence of from your assembler. And so you can actually use software or bioinformatics software to scan the contigs that you got from your assembler. You can predict where the restriction enzymes you used would cut the contig, and that would also hypothetically give you, computer-derived fragments that you should see on the optical map. And so you can kind of line up, essentially, you can use the predicted locations of the restriction enzyme cuts and match them to the actual cuts you observe on the optical mapping. And in that way, you can kind of try to figure out, okay, this contig, contig 1 kind of matches fragment 1 from the optical mapping, and so on and so on. And that, in some sense, helps you scaffold the genome.
And so the third approach you can use to order contigs, it's what's known as the chromosome conformation capture, or 3C methods. And so we'll talk about 3C methods in much more detail in the nuclear organization lecture, but I'll briefly review them here because they're relevant for ordering contigs as well. And so the idea is that even if two pieces of genomic DNA are far apart on the same chromosome, or even from different parts of different chromosomes, they can actually be in close physical proximity in the nucleus, because obviously the nucleus is three-dimensional. And so you can have folding and warping of DNA such that two pieces of chromosome that are far away in sequence are actually in close proximity. And so they can be in close proximity for a number of reasons. The first reason of which is that they may be kind of intentionally in close proximity. So you could imagine, for example, like a distal enhancer, as represented by the red ovals, can oftentimes be brought into close proximity to a transcription start site, represented by the blue ovals. And in that case, those two pieces of DNA representing the enhancer and TSS would be in close proximity. Another reason could be what's known as a bystander interaction. And so there, the idea is that you might have, for example, an enhancer contacting a TSS, for transcriptional regulation purposes. And obviously the enhancer and the promoter would be in close contact, but there may be also other neighboring fragments that are in close contact as well, just because they happen to be close in linear sequence to the enhancer and the promoter. Finally, you can also have just kind of random pieces of DNA being in close 3D proximity, just because of natural folding of the genome in the nucleus. And so here's a basic illustration of the 3C protocol. So the idea is that you start out with, for example, a pair of DNA fragments that could be far away on the same chromosome, for example. And what you can do is you can treat the cells such that pairs of fragments that are close in 3D space end up getting cross-linked to each other using chemicals like formaldehyde. And basically through the 3C protocol, which we'll discuss more in the nuclear organization lecture, you can essentially generate what's called a chimeric fragment, which is essentially a single DNA fragment that's composed of two pieces, where those two pieces are from different regions of the genome in this case. And so the idea is that when you sequence these chimeric fragments, the read that gets generated from this chimeric fragment will actually contain sequences from both fragment 1 and fragment 2, which again came from different parts of the genome. And so when you compare the sequence of this read back to the original genome sequence, then you can kind of figure out where fragment 1 and 2 came from, and you can therefore identify the fact that, at least in one cell, those two fragments that are far away on the linear chromosome were actually in close contact in 3D space. So here's a basic visualization of the set of all chimeric reads that you might sequence from a 3C experiment. And so the idea in this heat map on the left is that the first row and first column correspond to the first base on a chromosome, and the last row and last column also correspond to the last base on the same chromosome, for example. And the idea is that every single box in this heat map corresponds to a row and a column, which generally speaking are two different positions on the genome. And so when a box on this heat map is blue, that tells you that on average, the corresponding row and column, those two different fragments of the chromosome, don't really come in close 3D contact in your experiment. You didn't find many chimeric reads that originated from those two different positions on the chromosome. Whereas if you see dark red, for example, that tells you that you saw a lot of chimeric reads corresponding to that, corresponding to those two fragments, the row and the column, represented in this heat map. And so when you look at this diagram, basically the first thing you should see is a really dark kind of blackish line along the diagonal of this heat map. And what that tells you is that what the diagonal represents in this heat map is that, base number 1 is in close contact with base number 1, and base number 5 is in close contact with base number 5, and so on and so on. And that's kind of obvious, right? If you are in most contact with yourself, when you're talking about a linear piece of chromosome. And so the other prominent feature you should see on this heat map on the left is that there's kind of these dark red squares along the diagonal. And those are what are typically called TADs, or topologically-associated domains. And so what a TAD represents is basically the fact that entire domains or local contiguous regions on a chromosome tend to form these kind of essentially like a ball-like structure in the nucleus. And those ball-like structures are what are called TADs. And one of the functions of TADs is that they tend to facilitate a lot of, for example, like enhancer-promoter interactions within a TAD, and help prevent essentially enhancer-promoter interactions between like an enhancer and a promoter that are in two different TADs. And so TADs basically represent a lot of local interactions, potentially functional interactions of sequences within each TAD. And so we'll talk a lot more about TADs in the nuclear organization lecture, but basically for the purposes of ordering contigs, the important thing to note here is that if you happen to see, if there's a lot of enrichment of reads within each TAD, that means that essentially if you have a chimeric read that links two different fragments on the genome, then basically those two fragments would tend to be more likely to be within the same TAD, and therefore be kind of actually physically close in linear genome sequence than otherwise. So here's another visualization of what I was trying to explain earlier about how TADs help you order contigs. And so again, here's a representation of, say, 4 contigs that you might have sequenced and assembled. And suppose you're trying to use Hi-C to figure out how these contigs are actually ordered in linear chromosome space. So remember that at least looking at the heat map from the previous slide, we saw that these TADs, these boxes along the diagonal, basically tell you that there's a ton of reads corresponding to pairs of fragments that are actually somewhat close in linear DNA sequence space. And so the idea is that if you have 2 contigs and they are linked by a ton of chimeric reads from your Hi-C experiment, so by linked I mean that suppose you have a ton of chimeric reads where one fragment came from one contig and the other fragment on the same chimeric read came from another contig, then the more of those kind of reads you see, then probably the more likely that those 2 contigs come from the same TAD and therefore are actually close in linear DNA sequence space.
And so I'll just finish by saying that you can actually combine these different approaches in order to do a better job scaffolding. So you can imagine, for example, suppose you start out with doing PacBio sequencing to get some nice longer reads. And using PacBio, you can, for example, you could do an initial assembly of a genome to get contigs. And so you can take the PacBio contigs that you get, and you can use optical mapping to basically try to get an ordering on the contigs that you see from the assembler and get an initial scaffolding. And so your initial scaffolding might not be completely accurate because, again, your optical mapping procedure relies on you, selecting a good set of restriction enzymes that kind of nicely, evenly cover your genome and make cuts kind of as evenly distributed around your genome as possible. And that's not always possible because presumably you're doing this assembly because you don't know what the genome sequence looks like in the first place. And so selecting good restriction enzymes, may be actually pretty challenging. But once you use optical mapping to generate some kind of initial scaffolding, you can then do, like, for example, a high-C experiment, try to identify where are the TADs in my genome. So where are there, which regions of the genome should be in close proximity to each other by identifying TADs. And then you can kind of resolve the information that you get from the high-C contact maps and the information that you get from the initial scaffolding from your PacBio contigs plus your optical map. And so in the interests of starting a discussion during the live lecture, I'll leave you with some questions that you might want to think about. So number one, what do you think is the effect of having low sequence coverage of the genome on the number, length of contigs and your general difficulty of assembling contigs and scaffolds? Number two, what do you think defines a good set of restriction enzymes that you should use for optical mapping? And similarly, what do you think are properties of bad sets of restriction enzymes that you should try to avoid for using for optical mapping? And more generally, what is essentially the goal you're trying to, what are you essentially trying to optimize when you select a set of restriction enzymes for optical mapping, right? And so obviously different choices of restriction enzymes will lead to different distributions of cuts across your genome. And so, if your goal is to generate cuts that then maximally allow you to overlap restriction site cuts that you see on an optical map with those that you predict bioinformatically using your assembled genome, then what are the factors that you have to consider? Do you want to maximize the number of cuts that you make anywhere in the genome, or is making too many cuts bad? What about making too few cuts? And so basically I want you to think about what are the kind of design choices behind deciding which restriction enzymes should you be using for optical mapping.